I'm really looking forward to the zoom out as we go through the 3 days of the talk to see things at increasingly large levels of resolution as we get all the way out to populations and evolution. The Genome Browser is actually designed to try to simplify the large amount of data that is coming in. And Eric Lander told you earlier that the data are getting more and more complicated, and we're getting more and more dimensions to it. But what UCSC is trying to do is hang as much of it as possible on that linear sequence. Okay, it's a rows of G's, A's, T's, and C's, but there are a lot of things you can do to map to it, and what we try to do is hang anything we possibly can on the genome sequence. Okay, so the data flood that's coming in, we used to talk in the browser project, which by the way is 10 years old this month. The first code for the Genome Browser was checked in March of 2001. And we used to talk about the fire hose of data and how hard it was to handle the fire hose of data that's coming on. But about a year ago, the New York Times published this photograph, which really illustrates for you what it feels like to us to be standing in the face of what we used to call a fire hose. Okay, so we've got 80 organisms of sequenced animals. We've got 114 assemblies for those organisms, and we've got a whole host of information that has to be mapped to them. Okay, to start out with, there's 3 billion bases in the human genome alone. Multiply that times 80. We've got 114 assemblies, multiple assemblies for different organisms. We've got 33 million SNPs now, and the 1,000 Genome Project is adding more and more. We've got copy number variations. We've got the Decipher project, which attempts to hang phenotypes on individuals who have duplications or deletions. We've got the OMIM database, which has online Mendelian inheritance in man, which tries to associate functional information for each of the genes. We've got microarray expression. Each of the genes is turned on or turned down, turned up or turned down in various tissues. And then we've got multiple array probe sets, different platforms for CGH where users are running microarray information. And then in the human genome, we've got roughly 30,000 genes to map. Okay, so how do we handle that? Okay, well, can't handle it yet because the 1,000 Genomes Project, and there are a lot of people on Earth. Okay, so the amount of variation that the data set represents is huge. Oh yeah, and then beyond human, we've got the 10,000 Genomes Project, which is having a meeting as we speak in Santa Cruz right now to try to get together the sequences of all of the vertebrates that are possible from museum collections, endangered species, extinct species, and every important organism and every important clade to put together the tree of life at the sequence level. So what the Genome Browser does, which is genome.ucsc.edu, it's an open source system that's available to us thanks to the National Human Genome Research Institute. It displays massive amounts of genomic data for dozens of organisms, and it uses the simple paradigm of the sequence as a coordinate system. Okay, there's one reference assembly for each organism, although if you think about the earlier slide, it said there are 114 assemblies for 80 organisms, and that's because the reference assembly tends to mature, and as a new release comes out, we update the browser on a new organism, but we don't discard the old ones because a lot of people have annotations from their own laboratories on the older assemblies. So we have more assemblies than we have organisms, but it's still one reference assembly that happens to be maturing through time. We try to present a consistent paradigm so that when you're jumping from human to mouse to rat to fruit fly and back to human, you can do it and be comfortable in the interface so you can see that the navigation works the same way wherever you are. When we go to low resolution, Manuel talked about that a little bit earlier when he was talking about in one of his latest slides, one of his last slides about zooming in and zooming out. We try to give as much information as we can squeeze onto the screen at whatever resolution we are at. And if you zoom in, you get more. If you zoom out, you get less. You get more summary information. And in some cases, the summary information is actually coded in the tables for speed of display. For example, if you're going to condense 1,000 bases of DNA into a single pixel, you don't need to compute on 1,000 to get the value of that pixel because you know in advance what it's going to be. So we add rows in our tables for 1,000 and 1 million fold resolution so that we can jump to it very quickly. So as I said, we put in as much detail as can be displayed, and it's continuously variable from whole chromosome all the way down to individual base pair. It's pre-computed for speed where possible. In the earlier slide, I showed you that there were 71 million RNAs in GenBank. Well, we compute all those on a 1,000-node cluster on a run that takes 5 days to do. But then when you click on it, it doesn't take a few seconds or a few minutes to do the alignment because we know where the alignment is already. You click on an RNA, we show you the alignment because it's pre-computed. And it's interactive, dynamic, and inquiry-driven. The idea is that the user can decide what to display and what's important for his or her own viewpoint and what questions are being asked. And so you can provide the data sets for yourself and look at them at any resolution you like. Okay, so here's the whole view of chromosome 22, which is the smallest of the autosomes. And it's a 50 megabase chromosome, so we provide navigation at the top of the screen so you can see where you are relative to the chromosome. And here is a map of all of the genes that are known that map to chromosome 22. And as you can see, it's kind of a dense view. And you can see that the left arm, the short arm, has very little annotation on it all the way up to the centromere. There's essentially no genes mapped. You can expand the view so you can see individual genes by their names. The color density, the darkness of the color indicates the reliability of the prediction so that you can say, okay, this particular gene has very good annotation, and some of these have some kind of a code number. They haven't actually been named, but they meet the criteria for genes that have been algorithmically established. Okay, so it's possible to zoom in then by a simple drag and zoom process. You can drag across the toolbar at the top and then zoom into this particular region. And so you can see up here where you are, where you have zoomed in. And so these are all the genes that are present at that location. And so the paradigm is we show you as much as we can. So you can start to see the gene structure of some of the genes. There's a gene called LARGE over here. And you can see that the mRNAs that support this annotation map, the exons map as large tick marks. So these are the parts of the gene that actually become RNA. And the introns are in the low narrow band there, which is essentially part of the gene that is spliced out and thrown away in the transcription process. So you can already at this resolution see some of the structure of the gene itself. So to zoom in further, you can see that we're now down at the chromosomal level. We're at a single gene level here, which at the chromosomal level now is a tick mark, whereas the reverse was true earlier when we saw the whole chromosome. The genes were individual tick marks, so you can get a reference in either direction. So you can see here that this particular gene, MYH9, has a large number of exons. There's a large exon there. That's a region that encodes part of the protein. And you can actually see by the arrowheads which direction the transcription goes, so that the gene initiates on the right side of the screen and is transcribed and translated right to left in this case. Okay, so if you zoom in even further here to the 3' terminus of the gene, you can see that we have a half height box and a whole height box, which indicates the difference now between the transcription, which is the entire dark boxes, and the translation, which ends at this location. So the protein is truncated at this particular spot. So the farther in you zoom, the more information you get. And now if you zoom in on this exon here, it's the last exon of the gene. And the termination codon is shown here in red with an asterisk. Methionines, or start codons, are always shown in green, except they're not always start codons, but they are always shown in green, so you can tell which end of the gene you are on. And so the step down between the protein sequence and the untranslated region on the end is visible in the representation. You can also see that now that we've zoomed in this far, we're able to display the amino acid sequence along the base, and the information along the top of the screen shows you what the resolution is. We have a scale bar within each one of the pictures. We were at 1 megabase there, 50 kilobases, 1 kilobase, and now 50 bases here at this zoom level. Okay, so another type of data that we represent in the Genome Browser, this is the same gene in the same region shown at the top of the screen, and here I've turned on a track that shows comparative genomics data. So this is a 46-way alignment between human and 46 other vertebrates arranged in evolutionary order from the closest relatives of human, chimp and gorilla, down to the most distant vertebrates. Medaka and zebrafish are there are 5 fish at the bottom, and then lamprey, which is not really a vertebrate. It's a kind of an outlier It's a chordate. And so you can see that the region that is converted to protein is highly conserved, and you can see that in this wiggle plot, we call it. It's a graphical representation that shows that we have high conservation where the protein is, and then lower concentrations in the region around it. But I said earlier that we try to be an inquiry-driven interface, so you can see that there's a region here that's highly conserved, even though it's not in a coding region, which is the kind of thing that might get you to scratch your head and head to the laboratory and try to figure out why it is that this has been conserved through so much evolutionary time. If we zoom even further in this representation, we can see the individual amino acids where it is translated and the individual DNA bases where it is merely transcribed but not translated. And you can see the gaps and the insertions for each of the individual sequences, and you can see how the conservation works all the way down through evolutionary time. You can see up here the representation, the 6 and the 3 indicates that somewhere down in the display, there's an orange tick mark indicating a 6-base insertion at that location. The 3-base insertion is off the screen here. It's down here. It's off the screen of the blow-up. And there's also additional color cues. The green tick mark indicates this is the end of a piece of DNA that is sequenced, but the sequence breaks down even though there's some more sequence off-screen beyond that, so it's represented by ends. If there's no sequence available at all for this region, such as in the wallaby down here, then it's considered it's a complete blank on the screen. So we have a lot of visualization cues to try to give you a way to interpret the data as you browse. So if you zoom back out, you can see this is the region that was nearby that is conserved, and you can see that the conservation starts to break down in the intron as you get away from the exon, which is consistent with the idea that introns are thrown away and aren't evolutionarily constrained in the same way that an amino acid sequence would be constrained. If this protein is supposed to be functional, then its offspring are likely to have the same sequences through evolution, and the divergence is kept at a minimum. So as we continue to zoom out, you can see that the conservation really stands out, whereas the fish, lizard, zebra finch, and chicken, and so forth, the conservation is primarily in the exons where the protein is produced, and the introns it breaks down. And in this case, this particular view, you see that there's a big gap here in all of these 46 organisms relative to human. So you might wonder, what the heck is that gap? Why is there a big piece? Is it a display problem? Is it a construction problem? So you can turn on another track. You can turn on the repeat masker track, and you can find out that it turns out that there's a repeat in human. So there's a piece of DNA that it's inserted in human, one of the repeats that's lineage specific in humans, and that helps answer your question. So that tells you also something about the evolutionary timing of the insertion, because it's not present in chimpanzee. Okay, so you can ask and answer in a very short period of time that kind of biological question for any gene of interest. It's all database driven, and you can do the same zoom in or zoom out on any of the genes. Okay, so another one of our comparative genomics data tracks is what we call the net and chain track, where the entire genome, in this case of chimp, has been aligned to human, and then we represent the chromosome of chimp where the best match to human resides. And so you can see that a big chunk of chromosome 2 in humans, here's chromosome 2 in human, is a match to chromosome 12 of chimp, and the rest of it is a good match to chromosome 13 of chimp. Okay? So this shows the evolutionary footprint of a chromosome fusion event that took place in the lineage between human and chimp. And the timing of this event isn't well established, and it's not known if this may have been part of the speciation event that let humans head off on its own evolutionary direction, but it is a compelling argument that it might be involved in that process. So, in fact, it is so compelling, the association between these two chromosomes and human, that they've renamed chromosome 12 and 13, which used to be named in order of their size in the same way that human chromosomes are. They're now called chromosome 2A and 2B, which unfortunately has broken our software in the last two versions of human, because 2A and 2B both map to the same color, and it takes away the ability to show this nice evolutionary relationship. And I've got a change order ticket into the system for one of our engineers, the special case 2B, so it shows up as a different color. So I don't know about people from Europe, but people in the United States are involved in the process of arguing that evolution is a good explanation for things that have happened on Earth. And so whenever I speak to an audience of biologists or non-biologists, I try to include some kind of slide that shows that there's a lot of evidence for evolution. In addition to the fact that horses look like fossil horses, look like other fossil horses, there's a genetic and genomic footprint that also tells the story, and it's very compelling. For example, the introns and exons in some of the previous ones, and this is yet another one. Okay, so next generation sequencing data are part of that huge flood of information that's coming out of the pipelines now, and the graph that Eric Lander showed how fast the genomes are coming. We have the fact that the raw reads are coming out of the machines, then they're aligned with a tool called SAM tools, and they're put into files called SAM and BAM files, which are essentially huge pileups of short reads. So they can be mapped, they're mapped to the genome, and they may be as much as 80, 90, 100 bases. Some of them are much smaller, and they align in millions and millions of bases per, they talk about 20 million, 30 million, 40 million reads times the base length. So you wind up with this huge, huge file, okay? So there's a program called InSAM Tools that allows you to index this, which ultimately allows you to load the BAM and BAI files into a file on your own server, and you can upload it to the Genome Browser as a custom track. Okay, so here's a representation of some data that I obtained from Charles Nicolet at UC Davis. It's simply information that came off of one of their machines, and he didn't and wouldn't tell me what the information was, and I didn't really want to know because I was just interested in having something to display. And so what this shows is all the short reads that align to this particular region of chromosome, this 5 kb region of chromosome 21, and it shows you in red here where the data do not match the reference assembly. Okay, there are millions of well, dozens of genomes now available, and there will soon be thousands and millions of them. But most of them can be mapped, and there's no reason not to, is to map them to the main reference assembly. Okay, so here you can see the mismatches, so you can get down very quickly to see how the mismatches give you information about what's different between your genome and the reference assembly, which itself is actually not even one individual. Okay, so here's a zoom in to that one region on the previous slide. And so you can see here how the short reads line up. This is only 50 bases now, so your average read is shorter than 50 bases. And you can see that this particular sample has T's in this one base, at this one base location. And our representation here is just showing you the extent of the mapping and the places where the mapping differs from the base sequence. And so it turns out that this particular T, if we turn on the SNP track and we look at the 33 million single nucleotide polymorphisms that are available in dbSNP, so these are places where individual bases have shown up in a sequencing experiment and been deposited with NCBI. They're supposed to be at the level of 1 part per thousand in the population or more abundant. So they're benign SNPs. They're considered typical SNPs. And so this particular SNP has already been reported in the literature. Okay, here's a place where you have a heterozygous SNP in 2 places. Okay, where you have 3 of your 4 reads are A, and it turns out that A has also been reported in the database. And here, 5 of your 7 reads are G. So you can see at a glance if you have a heterozygote, although you may or may not choose to believe that 3 out of 5 is a heterozygote, or 3 out of 4 is a heterozygote, you might want to have greater read density. But you can see your data right away, and as you know, the eye is the best integrator of data there is. So you can see here how it looks. You can see here's an outlying one base. If you had a single read and you saw that, you might think, oh, I've got a mismatch here. But you've got a whole pile up of data that contradict that. Okay, and you can also see, because the BAM file contains depth information, it contains information about how good your alignment is. This particular mismatch is considered noise because it's in a region that wasn't very well, the quality score is fairly low. Okay, another data visualization method we're using on the browser is a way of taking multiple data tracks and superimposing them on each other. These are several data tracks that have something in common, and that is that they're involved in showing expression levels across the genome. And so you can see here that there's histone marks, places where the DNA is modified, hypersensitivity, places where the DNA is loose and available for access by an enzyme and by transcription machinery and so forth. And it comes from a number of different labs, including Brad Bernstein, who will speak a little bit later or next, I guess, John Stamatiopoulos at University of Washington and Barbara Wold at Caltech. So they generated the data, and then our data representation here says, okay, these different types of data show different looks at a similar story, and so you can superimpose them in a transparent color scheme, and so you can see not only how they pile up, but also where they don't pile up, because the colors bleed through then. Okay, so the next step is we're going to have data hubs where this huge pipeline of data, it is just too big to ship it all to Santa Cruz and have us make it available. So we're setting up a system where Ting Wong, who's chairing today's session at WashU in St. Louis, is going to be the first test bed for the data hub, where the data will live at WashU, and our software will make it possible for users to click on a box and view Ting's data on the UCSC Genome Browser, so that you'll be able to host your own data, report your data to us, we'll put a link to your data, and then you own the data, and people don't think it's ours, so it's buyer beware. They get your data from you instead of from us, and so we don't have to take so much responsibility for it, and that gives us higher throughput for the data. And here's a look at some of the types of data that will be shown in the epigenomics roadmap that Ting will be hosting, although this happens to come from the ENCODE project, which is a similar project. And coincidentally, this is also from Bernstein's lab from here at Mass Gen in Broad. And I actually put this slide together before I realized he was speaking after me. But here are some histone data that are, it's histone 3, lysine 4, trimethylation on a lymphoblastoid cell line. And you can see that they correlate very well. The pileup of that particular histone mark correlates very well with this divergent pair of promoters here, that it's at the 5' end of the gene. We're working to expand our capability to display differences between the new sequence and the reference assembly. And here's a picture of James Watson's sequence. And you can see the sequencing, the level of occurrence of the differences where Watson has a heterozygous gene here, or a heterozygous base. He's got a G and an A at this location where the reference assembly has an A. And then you can compare also by turning on the SNP track. So here's a place where Watson's SNP shows up in DB SNP. And in one type of display, if I show it over here, the G shows you what the ancestral allele is. So that's the allele in chimp, okay? And so you can see that one of Watson's alleles matches chimp and the other matches human, and no comment about that. We're also looking to find a useful display for three-dimensional data. There's some chromosome folding data that do not map very well to our essentially one-dimensional plot or two-dimensional plot. And we're looking for, at this meeting and other places, for creative ways to show three-dimensional data. I want to thank our sponsors, primarily the National Human Genome Research Institute. We also have money from several of the other National Institutes of Health, and also a consortium of Quantitative Biology 3. It's the 3 Bay Area universities of California, and also the Stimulus Fund sent some money to our bottom line. And this is the list of the talented programmers that I am privileged to work with every day. They've created what we think is a compelling view of the genome and access to all kinds of data that are essentially user-driven. We try to find good data sets and put them up there and let the user take off with them. And here's an old picture of the group. There's a new picture, including Ting when he was with our group. There's a new picture to be taken in 2 or 3 weeks' time when we're finally able to get them and the new people that have added to the project since that picture was taken all in the same place. So thank you very much for your attention. I don't know if we have time for questions or not.